In the last topic, we looked at synaptic plasticity and the idea that it might be a memory mechanism. So what I want to do in this topic is look at the early experimental evidence that this might be the case. In a later topic, we're going to look at more recent studies using new methodology, but for now we're going to start with the original studies. And this work really began with Eric Kandel's work in the 1960s. He was a psychiatrist interested, or still is, a psychiatrist interested in the connections between the brain and the mind. And he recognised that to really get at this question, it's necessary to have a simple experimental model that's looking at the really fundamental processes. So he turned to this animal called Aplysia californica, which is a type of slug, a sea slug, which lives in the waters off California, and it spends its days wandering around the ocean floor, grazing. It's quite a large animal, it's big enough to hold in two hands. And this animal has very simple behaviours, and to some extent they're very stereotyped, so almost like a reflex. In fact, some of them are reflexes, but some of them can be conditioned in both what's called non-associative conditioning and associative conditioning. So the conditioning paradigm that Kandel developed with these animals is to make use of a natural behaviour that they have called the gill withdrawal reflex. So as the animal is moving around, it gets touched by various things in its environment, fronds of seaweed and so on, and every now and then, of course, it's touched by something noxious. And it has a method for distinguishing the two. So if there's a non-noxious repeated stimulus, like being constantly wafted by water, then the animal will habituate to this, which means that it won't notice the repeated stimulation. But if there's something unexpected, then it will withdraw its gill protectively in what's called the gill withdrawal reflex. So the sensory inputs to this reflex are from a structure on its back called the siphon. And so if the siphon detects a mechanosensory stimulus, if it's unexpected, the gill will withdraw. So in the laboratory, this is really easy to reproduce in a very kind of stereotyped way. So what you can see in this slide on the left is a schematic of a laboratory paradigm of an aplysia, which is sitting in a tank, and there's a mechanosensory stimulus that can be applied to the siphon, and then the strength of the gill withdrawal response can be measured. Now, Kandel and his colleagues showed that if you repeatedly stimulate the siphon, then the gill withdrawal response will decrease. And they showed that this habituation of this reflex depends on a change in this chain of neurons. So you can see here the layout of the neurons, starting with sensory neurons at one end and motor neurons at the other end. So if you apply a single stimulus to the siphon, then the sensory neuron will release normal amounts of neurotransmitter, and the gill, the muscle in the gill will produce a response that results in the withdrawal. If you repeatedly apply that stimulus, then the sensory neuron starts to produce less and less neurotransmitter. So the strength of the conduction of that signal from the sensory input to the motor output is getting less. That's what habituation is. So it's a decrease in the strength of that synapse, so synaptic plasticity. So Kandel and his colleagues developed this paradigm to a very sophisticated extent. They showed that aplysia could be classically conditioned, so you can compare a novel stimulus with the unconditioned stimulus and show that the gill withdrawal response will occur to the novel stimulus, and they showed that, again, this depends on synaptic plasticity, and they worked out this very complicated sort of interaction between sensory neurons and interneurons. And if you're interested in this, it's a really it's a beautiful kind of set of papers that really have worked out this simple behaviour in quite a lot of detail. But we're kind of more interested in rodents ultimately, because we're mammals, and of course the LTP paradigm was developed for rodents to get at the question of mammalian memory. So how do we approach this question in mammals? So in the last lecture we met LTP, and we learned that it depends on the NMDA receptor, and the NMDA receptor has the property that if it's depolarized before the neurotransmitter binds, then the magnesium ion in the channel will be ejected and calcium ions will flow in. And so the neuron gets the message that there's been some type of confluence of events. So that's the sort of associative paradigm for the NMDA receptor. So it was found in the 1980s that there are some chemical compounds that can block the NMDA receptors and stop them from letting calcium ions in. And one of these is a drug called AP4, which was shown in laboratory studies to completely block LTP. So in a very influential study, Richard Morris and his colleagues applied this drug to the question of whether synaptic plasticity might underlie learning. So Morris had developed this method of studying spatial learning in rats, which we're going to look at in more detail later, called the water maze task. And you can see the water maze task in this slide here. It's basically a tank of water. It's 2 meters across, and if you put a rat in it, it'll swim around aimlessly in a bit of a panic until it finds that it stumbles upon a hidden platform, which is just below the surface of the water. So the animal can't see it or smell it or detect it in any way, but it kind of bangs into it, climbs onto it, and has escaped from the water and is very relieved about that. So if you repeat that a few times, put the animal in the pool and it'll swim around. Eventually, when you put it in the pool, it'll swim straight to where the platform is. So even though it can't see or smell the platform, it knows where it is. And the reason it can do that is because it can see in the room around the pool. It can see what's called the distal room cues. And so the animal is able to make a spatial map of the layout of the room and swim straight to the platform. And Morris showed in an earlier experiment that if you lesion the hippocampus, so if you damage it with electrical current or drugs or something, then the animal won't be able to find the platform anymore. It swims around aimlessly. So using this paradigm, he and his colleagues decided to test the LTP memory hypothesis. So if you block LTP, can you stop this learning, which we know depends on the hippocampus? So that's exactly what they did. They set up an infusion pump into the hippocampus of rats so that the rats were getting a continuous infusion of this drug AP4, which is APV, which we now call AP5. So continuously infuse this drug into the brain. And what they did was that they showed that this blocks LTP very effectively, and you can see that in the middle one of those panels. You can see that compared with the control conditions on either side, there's no LTP, so the evoked potentials didn't get any stronger in size. Notably, there were still evoked potentials, so this drug doesn't block normal synaptic transmission. It only blocks the increase in synaptic transmission. And then they tested animals in the water maze, and you can see in the middle panel that the animal that had the LTP blocked with the drug is searching aimlessly, very much like a rat with a hippocampal lesion. So that's really quite strong evidence for involvement of the NMDA receptor in learning, and by implication, involvement of LTP. Now, it's slightly indirect because you're infusing a drug, and drugs they tend to have non-specific properties, and you can't really control where the drug goes. So you can infuse it into the brain, but depending where you put it, it might go all over the brain, or it might go into a localized region, but you can't selectively apply it to just certain cells. And we'll come back later to new methods for actually achieving that, which have become very sophisticated. So another way of getting at the question of LTP in memory is to do it the other way around, is to say if these two things are associated, then if animals learn, we ought to be able to see LTP in the brain. And some early studies seem to suggest that indeed, if you measure evoked potentials when animals are learning a task, then the evoked potentials get larger. But this was kind of beset with all sorts of confounds. For example, it was found that the brains of animals warm slightly when they're learning, and if you warm the brain slightly, this results in an increase in evoked response size. So it was a little bit of an unsettled question. But then this experiment by the Moser group came along that seemed to provide pretty strong evidence for a spontaneous increase in the strength of synapses in the hippocampus in response to learning. So what they did was they used a paradigm called the inhibitory avoidance paradigm. So what this is it's an apparatus that has two chambers, one black and one white. And if you put a rat in the white compartment, normally it will walk through into the black compartment because rats instinctively avoid bright things and they go into the dark. That's where they feel safer. But with the inhibitory avoidance paradigm, what you do is when the rat has walked into the dark side, you give it some unpleasant experience, so typically a mild electric shock through the floor, so it gets a bit of a jump, and that's slightly unpleasant. So next time you put the rat in the white box, if it remembers that experience, it won't go into the black. It would rather be in the white, even though that's less attractive, because it remembers the foot shock. So it's a really good way of measuring how well the animal remembers a previous experience in a particular place. And the advantage of this method is that you know exactly the moment that you gave the foot shock, so you know exactly the moment that the increase in synaptic strength, if there is one, took place. So what they did was they applied this paradigm to animals, and they also had some control animals. So some animals were placed in the apparatus but just got a foot shock before they'd walked through to the black side. Some of them had the opportunity to walk through to the dark side of the box but didn't get a foot shock and so on. So they were able to kind of tease apart these different factors. And they showed that the animals that had the pairing of going into the dark box and receiving the foot shock showed a small but notable, significant increase in synaptic strength. And you can see that on the leftmost of those plots, you can see that the evoked responses have gotten slightly larger. So this is kind of like a naturally evoked form of LTP. So that's quite strong support for the learning memory hypothesis. The learning synaptic plasticity hypothesis, because these synapses have gotten spontaneously stronger. But it's quite a small increase compared with what you see if you induce electrical long-term potentiation. So if you remember back to those other slides, those increases typically like 100% or 200%, whereas these are just only a few percent. So what they did was they looked more closely at at where in the hippocampus these changes were happening. Because instead of using a single electrode to measure the synaptic strength, they were actually using a whole bunch of electrodes. So if you look at the photomicrograph there, you can see that there's an array of electrodes. And so this enabled them to sample evoked responses right across the hippocampus. And you can see the responses that they got in those heat plots. Each row of those heat plots is one of those electrodes. And the colour on the heat plot tells you the increase in evoked response that was measured by that electrode over time. And you can see the sort of four different conditions there. And if you look at the top condition, which is the pairing, so that was the leftmost of those graphs in the previous slide. So it's the pairing between the going into the dark box and the electric shock. And you can see that there's a lot of hot colours. So that reflects the general increase in strength of synapses. But you can also see that there's a lot of patchiness in that distribution. So not all of the electrodes saw an increase in strength. So the change happened in some places and not others. It was kind of heterogeneously distributed across the hippocampus. So that may be why it's been hard to see these natural effects in other types of paradigm, because they get diluted by the parts of the hippocampus that didn't change. But nevertheless, you can see that there are regions that had a very marked increase in synaptic strength. So that is very strong support for the synaptic plasticity memory hypothesis. Now, using interventions like drugs or lesions or whatever is quite crude, and as I mentioned, non-specific. So in recent years, new methods have come along that involve being able to actually manipulate the genes and produce much more targeted interventions in synaptic plasticity and memory. And in the next couple of sessions, we're going to look at those experiments.